Okay, so still in the middle of behavioral genetics here. Um, Announcements. So again, the perusal assignment is definitely available for you. I'm still working on this. I will give you at least a week. If I don't, I'll extend the due date. So don't worry about that. But the basically the Canvas-based assignment, that quiz-looking thing that isn't technically a quiz, that's still hidden. But it will be available, and I will give you at least a week to do it. I will adjust the due dates to make that happen. And the perusal assignment is out there. It's due next Sunday. So it's just a reading assignment. In this case, it's about the stingless, or sorry, not stingless, these sweat bees, halictid bees, which have become a model system for studying the evolution of eusociality because they're what we refer to as facultatively eusocial, which means that the same species in different ecological contexts will either express the social phenotype or express a solitary phenotype. And that makes them very interesting organisms to study. What are the ecological factors that go into this type of social behavior, and where did it evolve from, and so on and so forth. So that's kind of what that's about. So it's an evolutionary article where they argue for there being multiple origins of eusociality in these bees, and they use a phylogeny, and they talk about climate change and how climate change is involved in that, and so on and so forth. So I think it's an interesting article, and I encourage you to read it. So that's that. And where we left off is, this nature via nurture, or G by E, where we talked about, I'm going to use this phrase expression a lot. Sometimes we talk about gene expression or phenotypic expression, kind of just depending on what sort of fits in the sentence better. But the big thing is expression. And so expression is really, what is the phenotype, a phenotypic interpretation of the gene by way of the environment kind of blurring the interpretation. So expressing a phenotype is sort of this combination of the gene sort of pushing its way through the environment, and whatever washes out ends up being this phenotype. So we use this term a lot, then expression. And so last time we talked about how this modern synthesis of behavioral genetics had all of these kind of components, and we ended on molecular genetics. I didn't really get a whole lot of time to talk about molecular genetics. I want to spend just a little bit of time on that because there's some cool examples from that. So I would say the less cool case are these things having to do with what we call candidate genes. So candidate genes are like, for a long time, the promise of, and this isn't just for behavior, but for behavior, I think this is why it hasn't really been as useful. But the idea of a candidate gene is if you have a phenotype, but some traits that you're interested in, then you'd like to figure out what is the genetic basis of those traits. And so you can use molecular methods to try to test for that. So there are databases out there. They're called gene ontologies, or GEO databases, where people have sort of said, we know that this gene seems to be responsible for a heat shock response, or seems to be responsible for something to do with, a lot of times they're these metabolic the aspects of metabolic processes that go on in physiology. Rarely, sometimes they're annotated with behaviors. This seems to be a gene course that seems to make individuals aggregate and not. But a lot of times they're more these mechanistic. And I just want to emphasize that because when we talk about animal behavior, again, we have this proximate versus ultimate. So there are ants, for example, where people, there's never been a solitary ant in evolutionary history that we found. All ants have been social since the fossil record, as far as we know. And so in the laboratory, there are people that have created solitary ants that go wandering off and don't ever come back to the colony. And they've done that by knocking out a gene that has to do with how the ants smell. So you could call that a social gene, but if you looked it up in the database, like what does this gene do in a fruit fly, it would not have anything to do with sociality. It would have to do with olfaction. And so in this particular case, they created an ant that does not express this gene. And because of that, ends up behaving like a solitary insect, just walking around, foraging on its own, never coming home. So that's you could say that at the proximate mechanism, they've made the ant unable to smell a particular compound. But ultimately, they've gotten rid of the function of operating in a society. So it's not like the ant thinks, I want to be alone. And that's the kind of distinction of what we get between like proximate and ultimate. So a lot of times candidate genes are not something that codes for a complex social behavior. It codes for something that you might think of as physiological or sensory. But when you combine that with the system of other genes and its environmental context, you get complex behavior that comes out. Yeah. Well, a candidate gene is an idea of a single location on in an animal's genome that may have traits, that may have alleles that code for social traits. And so we need molecular methods like RNA knockouts, which I'll talk about here, are ways to test to see whether that hypothesis, to see whether that gene is responsible for sociality. A good way to test for that is to get rid of the expression of that gene or get rid of that allele and see if you get rid of the phenotype. And so that's what these methods, people have sort of been hunting through these databases and coming up with, yeah. Right. Well, so the idea here, and so this is a method, RNA knockouts. And so, as you know, hopefully know by now, DNA gets transcribed into RNA and then translated into proteins. And those proteins end up participating in complex metabolic dances with each other that end up allowing us to behave, to breathe, to smell, and all of these different things. Now, there are a lot of genes. And so the idea is here is you'd think, oh, I think the gene that is responsible for social behavior in ants is at this particular location. And so genes with, or ants with a particular allele, and maybe there aren't actually, maybe there's just one allele in the whole population of ants. Then the idea is, well, what if I just turn that off? What if I create an ant that doesn't have the ability to express that gene? And one way to do that are these so-called RNA knockouts. So it's a molecular method that will grab the RNA after it's been transcribed and prevent it from being translated. So it turns off the production of that protein. Yeah. I just know that there is a way to turn off individual genes. Well, that's just the term, is that a knockout is it's just you're knocking out that gene. No, yeah, the big thing I'm trying to communicate here is with older methods like RNA knockouts, RNA interference, small molecule RNA. There's all sorts of different ways to sort of knock out individual genes. And now we've got CRISPR, which has been in the news. And it's out of the scope of this class. We're going to go into the details of the CRISPR-Cas9 mechanism and how it works. But basically with CRISPR, you can get even crazier, where you can go in and find sequences of DNA and replace them with other sequences. So you have a way of engineering an animal that has different sets of genes. And that might mean just turning off a gene, or that might mean adding genes or replacing genes. Yeah. That's right. It turns off, the gene is in the genome, but it doesn't get expressed. Yeah. No, this is not a molecular course, but I just want you to know that it is possible to turn off genes. And in behavioral genetics, one of the things people try to do is figure out which individual genes are responsible for behavior. That's what I want you to know. But it hasn't been super successful. I mean, it has been, there's some great examples, but it turns out that behavior is really complex. It involves likely lots of genes. It kind of should, I think, make sense that an individual sequence probably doesn't have enough information to code for all of the different conditional ways that you can respond to something. Let's say in a mating interaction, courtship is probably not one allele, right? It's not like you've got Riz or you don't. It's probably, Riz is probably a combination of, it's probably a quantitative gene that has or quantitative trait that has a lot of different genes that are involved with that. So that's why this is sort of when these methods came out and people started studying non-behaviors, they were very successful, but they haven't been quite as successful trying to understand behavior because behavior is just a complex beast. In some cases, like those ant cases, there are a lot of promising results, but I think those are sort of exceptions to the general idea that boiling down behavior to a candidate gene approach is maybe hoping for a lot. All right, so that's sort of one branch of molecular genetics, is this candidate gene approach where you knock out candidates and see what happens to the behavior with different methods. But then there's epigenetics, and this is sort of a brave new world, and there's a lot of cool stuff that is going on with trying to understand how epigenetics works, and then the potential for how it interacts to create behavior. And so this is kind of like, if you look in the literature, G by E by E is something that you won't find many people using. You find a lot of the social scientists use it, and they talk about the early environment versus the current environment. And that, I think, is very much related to what I'm talking about here. So to a social scientist, V by E is like, not only how do you respond to your current environment, how do your current environment causes you to express behaviors, but how does your early childhood environment, how did that mold you in a plastic way into an entity that responds to the current environment? So social scientists are interested in the interaction between how you grew up and what environment is around you today. And that's where they get the G by E by E. I am introducing it in a kind of a more general way to just say it is the combination of your genes, and this is not my term, this is a real term, your epigenotype. And so there's actually different epigenotypes potentially for every cell in your body. And so what epigenetics provides us are non-genetic factors within an individual that regulate the expression of a gene. Similar to how we had epistasis, we said that was G by E. So if there's particular alleles, they change the interpretation of a gene that you're focusing on. Here, this is non-genetic effects. And so, and I'll give you some examples here in a second. So there are other things that are going on in the cell that if you were to read out someone's genome, read out their genotype, they might have the exact same genotype, but this cell is going to express that genotype differently than this other cell because this cell has a different epigenotype. It has different molecular factors attached to the genome that change the way the genome is interpreted. And that is sort of a much more modern way of thinking of this. So this is an example from NIH. I'm not going to focus on all of these, but the point I'm trying to make with this here is that there are a number of different mechanisms of, epigenetic mechanisms, and they're discovering more. And I'll talk about one that was discovered this week. Every day. So not necessarily every day, but we're learning a lot more about this because this is not something that we had the tools to really understand. So little things like, you've learned about the structure of your DNA. You may have learned about histones, which is, these little things that your DNA is wrapped around, how they wrap around those histones, who's wrapped and who's unwrapped. That affects the genes and how they're expressed. These little things called methyl groups. So DNA methylation, that's a big thing. So these are these little molecules that can be popped on to your DNA or popped off. And if they're popped on, then it basically turns off that gene. It knocks it out as if it's not there. And your physiology has the ability to make these modifications dynamically. And so if I focus in on that mechanism, like if you think about your different cells, they've all differentiated in, in these ways. You've got liver, you've got heart, you've got skin. And we know that cell fate, cell differentiation, DNA methylation, or epigenetics in general, but DNA methylation seems to play a huge role. So if you look at, for example, egg and sperm here at fertilization, there's actually a high amount of methylation on the DNA. But then during development, it largely gets stripped out. All those methyl groups get popped off, and then it rises up in the middle, and then it pops off again during development. By the time you're born, it rises up again, and so it appears as if your cells, as they're differentiating into the different types, they're using methylation and other epigenetic mechanisms as one of the ways to remember, I'm a heart cell, I'm a liver cell, I'm a skin cell. So to remember their fate. So they can say, all right, I'm going to turn off, I have the recipe for a pie, a cake, and a pizza. All of those recipes are in one book, but I'm going to, basically, tear out the pages of the things I'm not using, and that's kind of like what methylation is doing, is it sort of nulls out the recipes you're not using for that little kitchen. Every kitchen's got the same book, but this kitchen only makes pizzas and this kitchen only makes pies. And that is because they've sort of, blacked out the pages that they're not using. And that's kind of like what this sort of methylation is doing. Now, what's cool about this is that we're, again, this is an ant colony example, and this even has an ASU faculty member here, Jürgen Liebig, that now ants are, or social insects in general, eusocial insects, are emerging models for this term behavioral epigenetics. And so it's this idea of not only do epigenetics regulate the differentiation of cells, but maybe they also can regulate the different behaviors in individuals that have very similar genetic material. And an example of that is the social insects here. So this is a figure from that paper. And what they're basically talking about here is there are in ants, bees, there are these phases of development. So you've got a nurse early in life, and she becomes a forager later in life. And there is basically this threshold that starts very high when she's early in life and gets very low when she's late in life. And when there are certain nutritional transients, so the colony gets very, very hungry, then if it crosses that threshold, it will cause her to flip from being a nurse to a forager. Now, what's interesting is even if the nutritional stimulus goes away, she stays a forager. So her physiology, her behavior is now latched in to this new behavior, foraging, even though the nutritional stimulus has gone away. And there's a lot of evidence in looking at differences. So you can look at the genes that are different between this ant and that ant, but they're sisters. There's actually not a lot of genetic difference. But you can actually look at the epigenome of these two, and you can see, where do we see methylation? Where do we see different methylation, different patterns of methylation? And you find that in many cases, going from one type of worker to another is a shift in these methylation patterns. So they're sort of turning on a different set of recipes as they move, into a different stage of the restaurant's life. All the recipes were always there, but they sort of figured out there was a trigger that, okay, it's time to turn off these and turn on these. And that's what we mean by behavioral epigenetics. So, everything that I just said here, it goes to this question: how does a nurse switch to becoming a forager? That's the causal question that people studying this are trying to answer. Now, what type of causal question is this? If we've got these four types: function, phylogeny, ontogeny, or mechanism. What do you think? It has to fit into one of those four boxes. So we're talking about the how question. So that's one hint of a nurse becoming a forager. So what is the stuff that's going on inside a nurse that caused her to become a forager? And the time scale, I think, is also a nice hint here too. Yeah. That's a good question. So we've narrowed it down. Would it be mechanism or ontology? I think it is one of those two. Those are the kind of two how ones. Phylogeny, the time scale's way too big. That's like, how did ants develop division of labor? That would be a phylogenetic question. And then function, that's even more. That's like, that would be more of a why question. Why are they shifting from nursing to foraging? Why don't they just stay nurses? So I would say it's definitely either ontogeny or mechanism, but which one isn't? Yeah, it is a difficult distinction. I will say that both of these are mechanistic, for sure. I mean, we're effectively talking about developmental mechanisms or, your sort of everyday mechanisms. That's why I would say focus on this time scale. So we can still sort of differentiate. Like, I guess one difference is, you could imagine someone might ask, not how does a nurse switch to becoming a forager? They might ask, how does a nurse decide to feed a larvae? How does a forager decide to pick up a seed? That's a different type of how question. So with that in mind, does anybody feel like they've got a stronger stance? I think for me personally, this is one and only one. So I'm just seeing, does anybody else have a stronger stance? Yeah. Yeah, that's the way I would go. Is I would say, to me, this is an ontogenetic question, a developmental question, because it's talking about this longer term change. So we're talking about at this time scale. It's sort of up here where it's not what is the specific chemical cue that's causing this ant to walk over and take care of the brood? That to me would be a mechanistic question. It's saying, what are the factors that cause this ant to go through a shift in herself to become a different type of ant for all intents and purposes for the rest of her life? So to me, that's very much, thinking about, like, if we think about developmental questions from a cell level, we're talking about, at different points through the developmental cycle, you have to sort of ask, at what point does a cell divide? What point does it start to differentiate, and so on and so forth? And those, to me, are mechanistic because we're interested in the mechanism, but they're mechanisms of development. So, there you can the term mechanism is really difficult because in biology it's used all over the place. There is a group with a sort of a famous scientist that heads it down the street from here in the Biodesign Institute called the Center for Mechanisms of Evolution. A guy named Mike Lynch is the director of that. And Lynch's whole thing is about non-adaptive reasons for there to be evolution effectively. So he's really interested in phylogeny, but his whole center is about mechanisms of evolution. But it's a little complex because he's saying, like, what are the mechanistic constraints that would cause these evolutionary shifts? So, not what are the selective reasons why we would get something evolving from something else, but what are the mechanistic reasons? So it gets really complicated, the use of mechanism. But that's why I sort of try to look at time scale when you're talking about behavior. And in this case, we're talking about an ant that goes from one behavior and then shifts to another over a period of time. And it's not immediate. It's sort of more permanent. And that's why, to me, it's development. So there are questions about that. Okay, great, great. All right, so that's one of the cool things about epigenetics, this sort of cell differentiation or whatever. The other thing I want you to be familiar with is this notion of imprinting. And so imprinting is also typically done with methylation. This is often a target for sexual conflict between male and female in sexually reproductive species. So what imprinting is it's a way for parents to change whether their expression, whether their genes express in the offspring. So I'm not talking about alleles here. I'm really talking about the gene. So you can say that, that there are certain genes, like for example, this IGF-2, so insulin-like growth factor 2, that gene is one in which you get a copy from the male and female, from a paternal copy and a maternal copy. Only the paternal copy expresses itself. Regardless of what mutations or whatever, what alleles, whatever, only the maternal or the paternal copy expresses itself. So it's like the default is it for to be methylated and turned off, but in the sperm, in the production of sperm, it pops off that little methyl group and it says, no, actually, when this gene gets into the offspring, it's going to express itself. And then what this experiment shows here is if you, so we've got, this is the maternal expression of this IGF-2 receptor, which I'll get to in a second. And this is the paternal expression of the IGF-2 gene, not the receptor. And if you just express the male version without this female counterbalance, you get a giant mouse. Some people have done this. So they've created mice that they've done this experiment on. And without this other half that I'm talking about that compensates, the male gene, this male IGF-2 gene from the paternal side, causes the offspring to get really big. And it happens to be a major toll on the mother's body. So, perhaps not surprisingly, in this arms race, there is another gene for the IGF-2 receptor that is only the maternal one is expressed. So just as a reminder, in almost all of the somatic cells in your body, almost every gene, both alleles are expressed. There are important exceptions. The X chromosome is an example of this, and generally most genes, they've got two alleles, one from paternal allele, one maternal allele, both get expressed simultaneously. There can be interactions, but typically both get expressed. And so this is a weird thing where for these specific genes, these specific locations in the chromosome, or in the genome for the genotype there, the paternal one for this one is the only one that gets expressed, and the maternal one is the only one that gets expressed here. If you engineer these mice so that they don't have the paternal IGF-2 and they only have the maternal IGF-2R, then you get dwarf mice, really tiny mice. So you only get normally sized mice if both the paternal version and the maternal version are there, or both are absent. And that just goes on here. So this imprinting reflects a sort of a genetic or genomic arms race between the male interests and the female interests. The male interests want a big, healthy offspring, potentially at the cost of the female. The female want the offspring to survive, but the female wants to live to make another day. I say the female, but I'm really talking about the genes. We're taking kind of a selfish gene perspective here. So the male gene wants to be in a large, robust offspring. The female gene wants to be in a normally sized offspring. And so these two have sort of evolved as counterbalances to each other. So you end up getting normally sized offspring, with them both being present, which is probably the ancestral state. So probably, and now I'm making phylogenetic hypotheses here, but probably this was the ancestral mouse, and then maybe the male started to imprint to produce these bigger mice, And then females that figured out how to imprint for this gene here ended up bringing them back to normal. And now they're back in balance. So that's what we mean by imprinting, is it's ways in which epigenetics can make certain genes inherited from certain parents more likely to be expressed or more likely not to be expressed. Yeah. What do you mean by what amplifies? Like the mechanism of. Well, so in this case, like, and this is mammalian, by the way. This is you, me, everybody in this room. And so one of these common things in mammals is that males, for this gene that we all have in this room, you're expressing the IGF2 gene only from your paternal line, and you're expressing your IGF2R gene only from your maternal line. And that just happened over evolutionary history. So it is not a developmental thing. It's not a plastic thing. It's not like if you're malnourished, then you're likely to produce sperm that do the imprinting. All males produce sperm with that imprinting. All females produce eggs with that imprinting. And that's just a mammal thing. I'm sure there's a developmental biologist out there who would have interesting exceptions. Melissa Wilson would be a wonderful person. If you're interested in these topics, she is a world expert on sex and the evolution of sex and conflict. And a lot of it is about sex chromosomes, but this is totally in her wheelhouse. And I'm sure she would love to talk to you about this stuff. Yeah. This is certainly a route to dwarfism, but dwarfism, broadly speaking, I think has multiple causes. And I think what you're probably thinking of is a specific one, which I don't think is due to this. I think that's due to other parts. It's a great question. Yeah. IGF2R, that is the only expressed from the female, only on the maternal side. The paternal version is silenced. So when the sperm are produced, they said, we don't want this. So they turn it off. But when the eggs are produced, they say, we want this, they turn it on. Right. So when sperm are produced, they turn IGF2 on, but they turn off IGF2R. So sperm turn 2 on and 2R off. Eggs turn 2 off and 2R on. All right. Any other questions about that? So the term imprinting, it's sort of parent-specific expression. Mechanism that is as involved with that is methylation, typically, but that's the whole point of this epigenetics. And as we see, we're some crazy examples here. There are other cool mechanisms, non-genetic mechanisms that also can do this regulation. This is stuff that, like, you read about like Lamarck, for example. How many people remember who Lamarck is? Lamarckian inheritance, this theory that like Lamarck, had this idea that giraffes have long necks because a giraffe that had it kind of stretched and it got a little bit longer neck to reach certain leaves, and then its offspring had a slightly longer neck, and so on and so forth. And for a long time, people said, "Oh, those are just crazy ideas. That's just silly." But now we're finding these new mechanisms. Largely, they thought it's silly because we discovered these discrete units of inheritance called, genes. But now we're discovering things around the genes that actually do have the ability to do some of the things, not all of the things, but some of the things that Lamarck were talking about. And it's making us rethink, when we talk about G by E, all of those old ideas. All right. So this is an example that just came out earlier this week that I thought was pretty cool and relevant. And so this is, I don't want to read all of this stuff here, but the basic idea here is that this was in C. elegans, nematodes, little worms. But this is probably a conserved mechanism you'd find elsewhere as well. And what they found is that there are these amyloid proteins. So if you've heard about Alzheimer's, you've heard about amyloid plaque. They're these proteins that are created in different parts of your body, like your liver and stuff like that. It's a protein, it's not a nucleic acid. It is way down there. It's just floating around. It is not supposed to be something that we talk about in terms of inheritance or whatever. But what they found is that if they extract some of these proteins from so they have developmental phenotypes. So it doesn't matter what type of phenotype, but there's developmental phenotypes that vary across these worms. And if you extract some of these amyloid proteins from worm A that has phenotype A, and then you inject them into worm B that has phenotype B, and then you wait for worm B to have offspring, worm B's offspring will express worm A's phenotype. And so they didn't do any, methylation, histone modification, anything like that. None of the standard epigenetic stuff. They just injected proteins, and those proteins apparently have the ability to affect the development of these nematodes in a heritable way. And so they talk about it here that there are all of these familial developmental, I think they talk about it up here, numerous familial traits and disorders cannot be attributed here. Well, I think I'll get to I'll probably highlight that, I think, on the next slide. But they've sort of said, like, there's a bunch of things that we have no idea how they're being passed down, but they seem to stay in families and they seem to be developmental. This is one potential thing that could be doing that. This is a heritable developmental epigenetic modifier that potentially could be a target for some of these developmental disorders that don't have a genetic basis, but clearly look like they're heritable, which is kind of exciting. But the other kind of cool thing is they talk about this being a proteinaceous epigenetic memory when they talk about the why questions, the ultimate questions. Why does this exist? Well, they didn't do an experiment that does this, but this right here, this particular experiment, which is more mechanistic, it suggests the why question. Why does this exist? And they say that you can actually get changes in these proteins over the life history of a nematode that cause it to cause the development of its offspring to shift in a way that may be adaptive for that environment. That's Lamarck. That's the giraffe. This is a mechanism for that. And they're suggesting that in these nematodes that they can respond to the surroundings by changing the production. Like, remember, proteins, your genes code for proteins. You just ramp up a gene, you get more of the protein, you change what the fraction of proteins, and then some of that protein gets transferred to the offspring, and the offspring develops differently. It's built into a totally different nematode with totally different physiology, and that physiology is better adapted for that environment. That is crazy Lamarckian stuff. And it's not through methylation or histone modification or any of that stuff. It's through protein. Now, this, again, hot off the presses. I think this was September 2nd. This is when this was published in Nature Cell Biology. So, who knows if this will hold up. But this is the type of things people are starting to look into. And like I said, it suggests these favorable targets that potentially have these therapeutic benefits. So they say, again, there's a bunch of these familial traits and disorders that cannot be attributed to known heritable molecular factors. Here's one that is not a known heritable molecular factor, but appears to be heritable, appears to run in families, appears to be directly tied to development. So it's a target. We've gone from candidate genes to candidate proteins. So that's a little bit weird there. But the bigger picture I'm trying to say with this, so I don't need you, and there's a lot of questions about what do we need to know or whatever. You don't need to know about amyloid proteins. I want you to know about epigenetics. I want you to know that there is another mechanism inside an organism that affects the expression of genes. It's gene by environment by this other non-genetic thing that's inside the individual as well. That's what I want you to know. I want you to know that epigenetics exists, and it can take many forms. And this is one form. You don't need to know specifically about amyloids. You can forget that amyloids exist. That's fine. But I just think it's a really cool example. So the bigger picture here that I'm trying to get here is that, you know, if you think about this statement, what they're really saying is traits run in families. So that would suggest, and this gets into what we're going to talk about for the rest of this today, probably not the environment. But there are no known heritable molecular factors. All of the things we know that are inherited do not seem to have any heritability with respect to these traits. So we'll talk more about heritability and how to calculate it next time. But last time when we introduced the idea, remember, it was about populations. And it was about if you see variation in genes, you see it as their variation in phenotype. And if the genotype variation predicts the phenotype variation, it's heritable. Here they're saying the known variations in things that we can measure do not match the variations in these traits, despite these traits apparently running in families. So that suggests that we're missing something here, something epigenetic. And for them, they started looking for these proteins, and that helped answer it for them. But more broadly, about science in general, you know, science is often about explaining as much variance as you can. The genes for this particular phenotype, we can explain 40% of it by genes. We can, another 40% by environment, and we got 20% left over. And that 20%, man, that is where the fun lies, because that lets you then start asking new causal questions and start to, suggest new experiments. So that's what we do in science. We're constantly slicing variance like a chunk of rock where there is knowledge buried inside that rock, and we need to sculpt that rock away until eventually the knowledge just shows itself. But it's asymptotic. We'll never actually get to that pure form. We'll just keep chipping away more and more of that variance. And we're never going to explain 100% of the variance. But whatever variance is left over, that keeps us in business and allows us to tell us where to sculpt next, where to chisel out next. And again, for those scientists, it was looking at the proteins. For other people, it's looking at other things. But the question marks, man, that's exciting. Questions? Yeah, that's right. So the question, you ask questions about organisms. You would say, like, I notice that there is this in these mule deer or whatever, there's a variation in their phenotype. And what is that variation due to? Well, you could say, well, those mule deer live over there, and those mule deer live over there, and they never mix. And you can say, so it could be about their environment. Well, so let's take some of these mule deer, this relates very much to what we're talking about today, and move them over there and see what happens. And if they start behaving like those mule deer, then it is, we've explained some of it, but they probably won't be exactly like these mule deer. There'll be a little bit about that. So for that particular example, for these mule deer and that trait, then you've maybe explained that 60% of that seems to be intrinsic to the mule deer, and maybe 30% of it seems to be modulated by their environment. And there's 10% that is unexplained. And that 10% would then suggest, how to build your next research study to try to chop that 10% down. That's all I'm saying. So it's not constant across all organisms. For every particular question, that's what you're trying to assess variance, explain variance. And so in G by E, you say how much is in G, how much is in E, how much is left over for future work. All right, so making that a little bit more concrete, for the rest of this unit, that's kind of what we're focusing on, is what tools can we use to separate G and E so that we can get a better idea of how important the genes are for this particular behavioral phenotype, how important the environment is for this particular phenotype. And then after that, then we'll talk about actually then teasing out the genes. So which gene is which? All right, so with that in mind, are there any questions? Right, from epigenetics to the motivation for the rest of the unit. Yeah. The question marks are the unexplained variance. And so there's variance in behavior, and we like to know what are the origins of that variance. And it just doesn't come out of nowhere. So some of it is because the behavior is coded from genes. Some of it is because the behavior is a response, is involved with the environment's interaction with those genes. And any one study can is going to have noise in it. The study's not going to be perfect. And that question mark is often the things that the scientists at the time didn't think of. And so that's why the question mark suggests that, all right, what didn't we think of? What other things could we try? Maybe it's this other thing. Maybe all of those maybes are hypotheses. And those hypotheses can each be tested. So lecture 0, the first day you guys were in here, and I showed stotting behavior. And we spent, I don't know, 15 minutes where you just started saying, maybe it's this, maybe it's maybe. Those were all hypotheses. Now we could go back and say that was a developmental hypothesis, that was a mechanistic hypothesis, so on and so forth. Some of those hypotheses competed, some of them didn't. And the particular hypothesis I was looking for wasn't in that group, but all of those hypotheses were good. So that goes to show we could test all of those hypotheses and it would provide us some clarity, but the things we didn't think of are what's in the question marks. And eventually, once we think of them, we got to do an experiment and make those question marks smaller. That's science. Okay, great. So to the administration of it, let's do one of these. If I can find my mouse, okay. Short answer. And we'll do this guy. All right, so fill in the blank. Genetic blank or genomic blank is an epigenetic phenomenon that causes genes to be expressed or not, depending on whether they are inherited from the female or the male parent. So it sort of is lineage-specific expression. So what do we call that, the term? Let's see what we got results here. Okay, give this another couple of seconds here. And again, these are for completion or coherence, not correctness. All right, response is still rising a little bit. I'm gonna close it in 10 seconds. So if you're here and you want to be scored, then put something down. All right, so I'm gonna hit close now. All right, so everybody, almost everybody, a huge portion got it right. So 87% said imprinting, which is what I was looking for. Some other answers that came up, variants. We've been talking a lot about variants and genomic variant. I can see that it's an important term, but it's not specifically what we're looking for here. Inheritance is very generic. And allele, epigenetics, these are all things that, again, are much more sort of generic. The particular specific thing that I was looking for is imprinting, which is this term we use for exactly this phenomenon. Okay. Any questions about that? All right. Rid of the clicker here. Whoa. Okay. So this is what we're going to be talking about primarily today. What methods, what experimental methods can we use as animal behaviorists to help us separate genes from the environment when trying to explain behaviors? And then what we're going to sort of transition to for the last lecture of this unit, which will be Tuesday, is, all right, if we do think there's a genetic basis, there's a whole lot of genes. How do we figure out which genes are which? And so just generally the quantitative genetics of behavior, we're going to drill down a little bit more into that. We'll look at heritability. We'll see how we can calculate heritability, and we'll talk about QTLs and so on. So that'll be on Tuesday. But today, it's more about the behavioral methods for the rest of the class here. So Just a couple of examples, so it won't take that long to go through here. So one common method for separating G by E is something we call cross-fostering, the transfer of young between mothers. And this happens in nature. So there's a bunch of these natural experiments that happen. So these are from Australia, these cockatoos. So this is a gala or a rose-breasted cockatoo. Down there, they pretty much just say the gala. And it lives in the same area as a pink cockatoo. So first for bonus credit, well, not really bonus credit. Does anybody know the fancy name evolutionary or biologists use for living in the same area? Starts with an S. I heard this before in any of your classes. Sympatry. Anybody? Bueller? So sympatry. So these are sympatric species. They live in the same area. Not a term you need to know for this class. A good term to know though, because whenever that's one of the jargon words that we use. We say that they're sympatric. That means that they live in the same area. Sympatric. Sympatric. So these live in the same area, and they both lay eggs in these little holes that they try to find. These little mud holes. And they will often lay eggs in the same holes together. And they actually don't get aggressive toward each other until there's enough eggs and they start incubating. So that in itself is kind of a cool behavioral question, like the why question about that. Does anybody have any thoughts about that? Like, this isn't about cross-fostering, but just in general, why don't you immediately what's a hypothesis for why you wouldn't immediately be aggressive to another bird that's in the same nest area as you? Why do you wait until you've got a bunch of eggs and you're starting to incubate them? Yeah. Well, even before they start hatching, they'll start fighting. But while they're laying, they don't fight. So you'll have this cockatoo and this cockatoo in the same little mud hole together, and you'll have eggs of both cockatoos in there. And once you get enough of those eggs and they start incubating, at some point there's a trigger which causes a behavior to be released. That's the term we'll learn later. And that behavior that gets released is the aggression. But they don't immediately. It's not like when two cockatoos lay an egg, like immediately start thinking, I want to lay an egg right here. They don't immediately start fighting for that hole. They wait until there's enough eggs and there's an incubation, and then they become very aggressive. And when they become very aggressive, the pink one always wins. But that's another separate thing. So anybody have any ideas about why they don't just aggress initially? Yeah, in the back. So that's one hypothesis, that it's safety in numbers in the back there. Opportunity cost, it takes a lot of time to lay an egg. I don't really have time to be fighting off another cockatoo. So that the maximizing reproductive success, that suggests an ultimate explanation. And your ultimate explanation was that if they get into a fight, there's some probability they're going to die. And that would be bad because then it brings their reproductive success to zero. Does that sound right? These are all good hypotheses. Anybody else? All of these, I would say, are functional hypotheses, which makes sense. I asked a why question, right? It was a functional question. I could have asked an evolutionary question about like, how did this evolve from others or whatever. I actually don't know the answer. We could sit here and brainstorm for the rest of the time to figure out how we could build an experiment to test those out. I think, me personally, I like that last answer about aggression. To me, is really costly. People always make a big deal about honeybees having barbed stingers. Like, how often, like, if you got stung by a wasp, I mean, you respond to that, right? Like, if there's a wasp on you and you get stung, you probably are going to try to smack that wasp, right? I mean, you're like, it's just, so it doesn't really matter that there's a barbed stinger in me from a honeybee. If you've gone out and aggressed, you are bringing it on. That really steps it up. It's hard to get out of an aggressive situation. And my guess, totally a guess, totally a hypothesis, very much along the lines of that last one. But I like both of the other answers too. But my personal guess is that it's just too costly to go to aggression until you have enough of an investment. And once you have a bunch of eggs and you're incubating, now you have a bunch of investment. And at that point, it's worthwhile because you've kind of got enough momentum that it shifts the balance in the other way. But I really like the idea too that it might just take time, or if they have natural predators, there can be these sort of nurse effects of having these others around. All great ideas. Again, I don't know the right answer. I think somebody does. So if you're interested, you could probably look it up. But that happens to be one of the things, is that they will get into fight, and this one will always win. Now, what's cool about it is that this one doesn't realize that it's got eggs from this one in its nest, and it raises them. It's a natural so-called cross-fostering experiment. So that the pink cockatoo will raise the chicks of the rose-breasted cockatoo. And then what people have done is study phenotypes of the resulting rose-breasted cockatoos that were raised by the pink cockatoo. One of the things they found is that the rose-breasted cockatoo, so there are these different calls, and I guess it's animal behavior, we should talk about this, is that birds have begging calls. So the chicks have a call that it seems to be to bring a parent over to feed them or to let them know that they're hungry. They have alarm calls. So if there's a predator around, then they let them know the alarm. And then they have contact calls. And that's supposed to say pink cockatoos. And contact calls are like just the general noises that you hear when you walk around. You're like, oh, there's just birds being noisy. And those seem to be to help maintain social cohesion. So birds hear themselves, and then they know they can, either aggregate or disaggregate based on listening to these contact calls. And what they find is that the rose-breasted cockatoos raised with the pink cockatoos produce alarm calls and begging calls that sound just like rose-breasted cockatoos. But their contact calls sound just like pink cockatoos. So what's the G by E story here? What do we interpret about the different phenotypes that we're talking about here? Any thoughts? Why is that a useful and cool result? What does that hint at? Yeah. Yeah, so that's a great, so the two of these, begging and alarm, are more affected by so you said nurture or nature for this one? Nurture. So these ones, this is the gala. Did you mean, because it's sort of funny how these two calls are like their normal calls. So yeah, flip it around. Yeah. So, but yeah, generally, yeah. So the idea here is that potentially what this supports is that these two calls are primarily encoded in the genes. The gene is the primary thing driving those two calls, because you changed the environment and it didn't learn the call from the mother or the father. It learned, it was already in the genome. But this call, that suggests it's more determined by the environment, because we've shifted it into the nest of the pink cockatoo and its call shifted that way. Now, there is a caveat here that I'm going to highlight, but I'm going to wait until this next example for us to talk about that caveat. So here's another example, again, from birds. Animal behaviorists love birds for some reason. So a lot of great examples of birds are really good model organisms. This is a great tit. This is a Eurasian blue tit. So these are two birds that you'll find all over Europe. So this isn't a natural cross-fostering experiment. This is an experiment where they've done it in the laboratory. So, well, actually, in this case, they've done it in a field, but they manually did it. So they were studying the same sort of thing, where they took eggs from a great tit, and they took the control case. Eggs from a great tit are raised by a great tit. In the treatment case, eggs from a great tit are raised by a blue tit. Likewise, they did the other way around too. The control case for the blue tit is eggs from the blue tit are raised by a blue tit. Eggs from in the treatment case, eggs from the blue tit are raised by the great tit. And then they looked at the calls. And what they found is that, so up top here, and there's a similar story down here, but for time, I'll probably just focus on this top one here. These two bars here are for the blue tit. This is the control, and this is the treatment, the cross-fostered. Over here, these two bars are the great tit. So this is the control, and this is the cross-fostered. So what that means here is that this bar represents a blue tit being raised by a blue tit. This here is a blue tit being raised by a great tit. Likewise, this great tit, this one here is raised by a great tit, and this great tit was raised by a blue tit. And so, and these are number of notes. And so this is a measure of complexity of the song. And what they saw is that with the blue tit, it doesn't really matter who raised you. But with the great tit, if you get cross-fostered and are raised by a blue tit, then in that case, your song changes. So that sort of suggests that maybe with the blue tit, songs are more genetically determined, but with the great tit, they're more environmentally determined. That said, and I don't want to go into all the details here, they did find that in all of the cross-fostering experiments, there was a so-called heterospecific song. So there was an intermediate song that was produced in both cross-fostering cases. So there is a little bit of environment in both, but it's primarily gene, at least that's our interpretation, in the blue tit, and it's much more environment in the great tit. Do we see that example there? You see this cleverness about switching, taking genetics and putting them in different environments. Devious. And it helps us get these interesting examples out of here. So questions about that? Does that seem clear? Okay. Now, caveat. This is the same caveat with the last one. These researchers did it in natural nests in the field. So remember I said these are sympatric. So you have great tits and blue tits that are just next to each other. So what's the potential problem there with a cross-fostering experiment when you do it in the field when both types of birds are still around? Any thoughts? Yeah. All right, so the thought was there that even though you took a blue tit and you put them in a great tit nest, there still might be blue tits around that you could hear, even if they're not your parents. Yeah. Was there a similar comment in the back there? Yeah. Yeah, and that's an issue with this experiment. That would be, if you were a reviewer, you would say, interesting results. They tell us some interesting things here. It didn't quite disentangle that. In a next version of this, they might actually make you to go back and redo the experiment. But you can imagine doing this in the lab in a more controlled experiment where that you could definitely control the experience. And like I said, it's the same deal with the cockatoos. They're sympatric, so even though there's cross-fostering, there still is your species nearby that you can hear from. Cross-fostering also, crazy enough, can be done inside the womb. And so this is kind of a fun model system, is that you've got these mice, for example, here, and there's these two backgrounds here. So notice this mouse expresses this called ball, basically brown versus white. And so they've taken an embryo, which is the brown, which has it has the gene, it has the B6 gene. I don't want to equate the gene with the phenotype, because that's the whole point, is we're trying to figure out where does the phenotype come from, the gene or the environment. All we know is that the embryo's got the B6 allele. So there are two alleles, there's B6 and there's bulb. They could be at the same spot in this gene. So we're going to take the B6 embryo and we're going to put it in a B6 recipient and in a bulb recipient. Let it come to term. Once it's delivered, then we're going to take it and we're either going to let it be raised by a B6 mom or a bulb mom. Same thing over here. So now we've got one embryo with B6 that has both a different, it's been cross-fostered in the womb, and then it's then been cross-fostered throughout its development out of the womb. And so they have this kind of interesting result that cross-fostered embryos behave like the recipient strain of mice. But if you perform the cross-fostering after birth, then the mice behave like their biological parents. So to me, that if I read that right, that seems like the womb is pretty important there. So if you're looking down here, this B6 goes to B6. If it's raised by the white one, the bulb, it's still B6. If this B6 is going through the bulb and it's raised by the brown one, it's B6. If it's raised by the white one, it's bulb. So you can kind of do all of these different combinations and then try to tease out where there's the gene and where is there in the environment. But the big thing that I want to show here is just on the methods case, if, in certain organisms, you can actually do this interesting cross-fostering. There's crazy things that are sort of related to this too. In honeybees, you can take the ovaries out of one bee and place them in another bee. And when you do that, she'll switch to the job of the donor of those ovaries. So if she was a nurse and you transplant that into a forager, then the forager who gets the nurse ovaries starts being a nurse again. So, that's also like, a lot of interesting sort of things that brings up. But anyways, I want to get to, another two quick examples here. So I'm not going to go into as much detail here. Twin studies. This has been speaking of, trying to understand the role of genetics by using the womb as a laboratory. There are a bunch of times that, at least in humans, you have monozygotic twins or dizygotic twins. So monozygotic twins, identical twins, they develop and they have basically the same genes. Dizygotic, they may as well be born at different times, but they happen to share a womb. And so what twin studies do is they look for cases where you can have the same mother, for example, or the same background environment, and you try to compare outcomes between these two different groups that have the same gene and groups that don't. And if you have monozygotic twins that look more alike, then we would think that would mean the genes are probably more important. But if the monozygotic twins don't look any more alike than the dizygotic twins, then that would suggest that the environment is shaping that behavior. So that's one of the cool things about these twin studies, is that you can sort of study the differences between the twins and try to get an idea of this G by E. Now, the downside of these twin studies is that in non-human animals, it's really hard to tell if you've got identical twins. So you have to do additional genetic testing that you may not have to do in human experiments. So it's not the most practical thing. I wanted to cover one more thing here, and that's common gardening. So now that we've seen all of those, I just want to see that based on this, so the term we have here is common gardening or transplant experiment. Anybody have a guess as to what you do in a common garden method? And the hint also being that it's called a transplant experiment. And this is particularly valuable when studying ecotypes. All right. I won't torture you then. So the idea here is that, if you have ecotypes, remember these are members of a species that have been separated from each other and now express a phenotype that may be tailored to that environment. Well, there's the question of what is that ecotype genetic or is it environmental? Are they just filtering through the environment and that's why they're behaving differently, or have their genes actually changed? Well, a great way to do that is to just take those individuals from those different areas and put them in a so-called common garden. So let them develop, for example, in a common environment that you can build and maybe control. And then you see, do the differences persist or do they go away? If they persist, probably the genes. If they go away, then it's probably the environment. So you could argue that a lot of the methods that I just talked about are special forms of common garden, but this is very broad, and you see this in a lot of different behavioral experiments. All right, so that was all I wanted to cover today about this G by E and disentangling those. I want to get into more, and I've got a question for you, and I want to get into more of the quantitative stuff on Tuesday, and so that's kind of the goal there. Then we'll be done with behavioral genetics. So remember, perusal and Canvas. If I don't get the Canvas assignment, you'll definitely get a week to do it, and I'll extend the deadlines to make that happen. And otherwise, I'll leave you with this. So I'm going to start the iClunker multiple choice. So what is the name, terminology here, the most specific name for the method that places the young into the care of another? So the method that places the young of one into the care of, and I think I was supposed to say care of another. So we're moving young from one, let's say, mother to another mother. What method is that? Otherwise, have a good weekend. See you next week. Yeah, let me stop the recording here. I will eventually hear once things settle out. But you can submit. I think you think you're good. Uh-huh. That was the